Hey, assalamu alaikum. Today we are going to discuss about the nucleic acid. What are the nucleic acid? What is the history of nucleic acid and what are the structure and function of DNA? In the next we are going to learn about the structure and function of RNA, then the difference between DNA and RNA, and after that the chromatin and DNA topology. First of all, we are moving towards the history of nucleic acid. In the 1859, Charles Darwin, Theory of Evolution, humans shared a common ancestor with apes, which is a chimpanzee. This is a theory was from the Charles Darwin, which states that the human is from the Japanese, as they have a common ancestor with the Japanese. And after that, in John George Mendel in 1822 to 1884, which is also called the father of genetics, he worked on a pea plant and discovered the fundamental law of inheritance. He also explained the genes come in a pair, one from each parent, and after that there was a Mendel's law of heredity, are usually stated as the law of segregation, and the next one is the law of independent assortment, and the third one is the law of dominance. These all the laws we also discuss in our FSC level. And after that he published his results in 1865. Mendel's work was not appreciated in his time until 1900. After the rediscovery of his laws in 1900, his experimental results were understood. Now, in 1800, in 18s, John Friedrich Mischer in 1844 to 1895, which is the date of birth and the date of death, Swiss physician and a biologist. He was the first scientist to isolate nucleic acid. In 1869, Mischer isolated a material rich in phosphorus from the cell from human WBC, or you can say white blood cells. He found a substance that has unusual chemical properties unlike the proteins. He also found that the substance has a very high phosphorus content and was resistant to protein digestion. He called as nuclein. Despite this, it took more than 50 years for a wider scientific community to appreciate his work. We are discussing all the history of nucleic acid. Now, in 19s, Rosalind Isles Franklin, which was born in 25th July 1920 and death in 16th of April 1958, was an English chemist. There is a picture of her and also her work. He was an English chemist and an X-ray crystallographer. Her work was central to the understanding of molecular structure of DNA, which is a deoxyribonucleic acid, RNA, which is ribonucleic acid, virus, coal, and graphite. In 1952, she crystallized a molecule of DNA from the X-ray diffraction images. She demonstrated that DNA contained a regularly repeating helical structure. The image allowed a precise calculations of molecule spacing in the DNA. This was basically the image. Now, in also 19s, Marie Curie, Hughes, Franklin and Wilkins, which is from 1960 to 2004, he was a New Zealand-born British physician and a molecular biologist, and he also got a Nobel Prize. His research contributed to the scientific understanding of phosphorescence, isotope separation, optical microscopy, and X-ray diffraction. Marie Curie, Wilkins, and Franklin used X-ray technique to the first photograph of DNA. The clear image they provided, which pointed to the double helical structure. Now, 1953, James Johnston and Franklin developed a double helix structure of DNA. Using available X-ray data and model building, They were able to solve a puzzle that had mystified scientists for decades. They published the paper in Nature in April 25 in 1953, and in 1962 they were awarded the Nobel Prize for Physiology or Medicine along with Marie Wilkins. Despite the fact that her photographs have been critical to Watson and Crick's solution, Rosalind Franklin was not honored as only 3 scientists could share the prize. Now I have completed the history of nucleic acid. Now move towards the 6 amazing but true facts about the DNA. What is the 6 amazing facts? Number 1 is our DNA could stretch from Earth to the Sun and back 600 times. If unwound and linked together, the strands of DNA in each of our cells would be of 6 feet long. With 100 trillion cells in our body, that means if all of our DNA were put end to end, it would stretch over 110 billion miles. That's hundreds of round trips around the sun. So this is the first amazing fact of DNA. Now, what is the second amazing fact of DNA is we all are 99.9% alike. Of the 3 billion base pairs in the human genome, only 0.1% are unique to us, while that 0.1% is still what make us unique. This is basically very amazing. Now, it means that we all are more similar than we are different. Now, the number third 6 amazing facts of DNA is gene make up only about 3% of your DNA. Genes are short segmented of DNA, but not all the DNA is gene. All told, genes are only about 1 to 3% of DNA. The rest of your DNA controls the activity of your genes. Now, what is the fourth amazing fact is a DNA test can reveal you are more Irish than your sibling. This is true for any of over 350 regions covered by ancestry DNA test. What is the number fifth fact of DNA, which is amazing? The human genome contains 3 billion base pairs of DNA. DNA molecules are shaped like twisted ladders. The rungs on the ladder are made up of bases which are adenine labeled as A, cytosine labeled as C, guanine labeled as G, and thymine labeled as T. Look together in pairs with hydrogen bonds. So there is a hydrogen bond between the bases. The really cool part is if they pair up in a very specific way, A always pair up with a T and C always pair with a G. So the shortcut of learning is at GC. So A always come with T and C always come with G. Now, what is the last amazing fact, though basically not last but not the least, is your DNA could link you to place you would never imagine. Genetic has the power to tell you the things you never dream of knowing from just the DNA in your saliva. With the ancestry DNA test, for example, you can find out which world region your ancestor may have lived in 100 to 1000 of years ago, what bonded them over generation, and why and where they migrated, all from your DNA. So in the next part, we are going to discuss about what is the difference between the cells, prokaryote and eukaryotes.